My name is Tamara Lipsey, and Eric and I have been hard at work in getting the dissolved oxygen and temperature meters ready for everyone to use. On the left is one of our types of meters. In the middle is someone taking dissolved oxygen and temperature readings. And on the right is Eric helping me measure out the cables and the depths for you all. As I said, my name is Tamara Lipsey. I'm an aquatic biologist with the Michigan Department of Environment, Great Lakes, and Energy. I've been a biologist for about 20 years, and you can see in the upper left, that's me taking Secchi disc readings, and we take phosphorus readings and dissolved oxygen and temperature readings as well. So today we're going to talk about dissolved oxygen temperature and why it's important first. Then we'll go over the MiCorps CLMP program overview for dissolved oxygen and temperature. We'll go over the equipment, we'll go over the procedure, and then we'll go over a couple of end of year reminders. So what do dissolved oxygen and temperature measure? Well, obviously, temperature measures how warm or cold the water is. And dissolved oxygen is just what it sounds like. It's the amount of oxygen that's dissolved in the water and that is available for aquatic organisms to use, like fish and other aquatic organisms. But why is DO and temperature important? Well, it affects and it's affected by many physical, chemical, and biological components of a lake. And we're going to return to this question a little while later after we learn some more about the properties of dissolved oxygen and temperature. So first I want to talk about thermal stratification. By measuring temperature, you can determine how your lake stratifies. And this is a diagram of how many lakes stratified during the summer in Michigan. The upper layer is called the epilinion, and this area is very stable in temperature, and it includes that shoreline area right against your riparian area. The upper layer is warmed by the sun and the air temperature. And then when you get a little bit deeper, maybe if you're swimming, it might be around your toe area, you'll notice that there's a rapid decrease in the temperature. This middle layer is called the metalimnion, and that area where the temperature declines pretty rapidly is called the thermocline. Then you have a cold bottom zone called the hypolimnion, and this layer remains cold throughout the season. So to learn a little bit about why our lakes stratify into these layers, we're going to learn a little bit about water and the density of water and how it changes with temperature.
Imagine you're swimming at the lake on a hot summer day, and your friend says, "Hey, I bet you can't touch the bottom." And you're like, "I bet you I can." So you take a deep breath and you swim down and down and down, and suddenly it gets cold and you stop. What is this? What's going on? You've just experienced something that's hugely important to everything that goes on in the lake. It's called thermal stratification. After you get back to the surface and catch your breath, you think, why is it warm up here but cold below? Is it always this way? Let's run an experiment and find out. What we have here is a model of a lake cut in half down the middle. We've taken all the warm water, dyed red, and moved it to one side. The cold water, dyed blue, is moved to the other side. In the middle is this barrier that keeps the sides separate. Now let's remove the barrier and watch what happens. As you can see, there's a lot of movement between the two layers as they come together. But look, the warm water has settled out on top. Let's try the experiment again. This time we put the cold water on the left and the warm water on the right, just to see if it makes a difference. Once again, the warm water settles out on top. What's going on? To answer that, we need to take a look at the physical principle driving the whole show: density. Density is the amount of stuff in a given volume. In our case, that stuff is water, and that volume is this glass beaker. This beaker is filled to the brim with our cold water. Let's see how much it weighs. $166.2 grams. Now we've emptied the cold water and refilled the beaker with the warm water. And the warm water weighs in at 165.1 grams, a little bit less than the cold water. We can calculate the density of these two samples by dividing the mass by the volume. First, we need to correct our previous measurement by subtracting out the mass of the empty beaker. This gives us 116.2 grams for the cold water and 115.1 grams for the warm water. Now we divide by the volume of the beaker to get a cold water density of 1.00 grams per milliliter and a warm water density of 0.99 grams per milliliter. In reality, it's a very small difference, but that small difference has a big impact. It allows the warm water to essentially float on top of the cold water, and it keeps the two layers from mixing together. So what affects the temperature and in turn the density of the water in the lake? You might think the temperature of the air surrounding the lake is the answer, but really the biggest impact comes from the sun. As sunlight enters a lake, it warms the surface water, making it less dense and lighter. As the surface water begins to float on the cold water below, a transition zone forms where it changes from warm to cold very quickly. We call that the thermocline. Remember, thermal stratification in lakes is as ordinary as the changing of the seasons, and in fact, it does change with the seasons. But we'll save that for another time. For now, know that warm water, cold water, and a thing called density set the stage for the behavior of, well, really everything that goes on in a lake. It's very important.
So hopefully you all enjoyed that video. I appreciate seeing a visual of what's happening in the lake, and that stratification is so important, as I said, to many functions, ecological functions within the lake. Now they mentioned that things change with seasons, and that's what we're going to talk about now. So here's the summer, where you have your apolimnion, which is that warm layer, the thermocline, and the hypolimnion, where the cold water is much more dense and at the bottom of the lake. Well, as the air temperature drops in the fall and the sun gets lower in the horizon, the temperature in the lake begins to fall. And then the winds that come up in the fall mix with the air and add to this cooling effect, along with not having as much sunlight in the fall. So then that cold water begins to sink, and it pushes all the warm water that's below up to the surface. And this process is called lake turnover. And as that warm water is rising to the surface, the nutrients that are in the bottom of the lake will also mix upward. So then when we get into summer, the air temperature continues to get colder and colder. Oops. And as that layer of ice begins to form, it insulates the water underneath. And although that layer is very thin and the difference in temperature can be, just a mere couple degrees, that helps the fish be able to survive over the winter. Then we get into spring, and that ice melts. And as it melts, it'll reach that 4 degrees Celsius, which is when water is at its densest, and it will sink to the bottom. So that slightly warmer water will rise to the surface again. And this is called spring turnover. And again, the nutrients that are in the bottom of the lake and the oxygen levels are mixed thoroughly during the spring turnover time. And so that's why we have you sample a spring phosphorus sample, because you're getting the entire water column's level of phosphorus. Okay, so we are going to move on to dissolved oxygen and the importance of dissolved oxygen. I can pause for a minute and see if there's any questions.
Hi, Tamara. Yes, there's one question in the chat from Terry, and she asks, when there is not ice, because we barely had ice this winter on our lake, is there significantly less turnover? And if so, what practical implications are there?
I wouldn't say that there's significantly less turnover. The lake is still cooling enough in the fall and at those times during the winter where it was cold to mix the water. And then in the spring, it's going to be uniform in temperature. But that is why we want you to get out there so soon after the ice melts, because it will start to warm quickly. So in years like this year, as soon as we know the weather's going to start getting warmer, which was usually in March, that's when we have you do the spring sampling because of that turnover.
Thanks, Tamara. That's the only question in the chat.
Okay. I'll wait a few seconds and see if anybody wants to unmute their mic. Okay, so we'll move on to dissolved oxygen and how it interacts with temperature and how it's also very important to the ecology of the lake.
Imagine a world where we weren't surrounded by air. Without the oxygen in the air that we breathe, we wouldn't survive very long. But what about animals underwater? They need oxygen too. Where do they get it? The answer is from the water, because water actually also contains dissolved oxygen. Dissolved oxygen is just what it sounds like. It's oxygen that's dissolved in water. But what does it mean for something to dissolve? If you've ever mixed sugar and water, you've noticed that the sugar seems to disappear. But what really happens is that the molecules of water mix with and surround the sugar molecules, breaking up the large sugar granules to make a sugar-water mixture. More specifically, since the parts of the mixture are spread out equally and evenly everywhere, we can call this mixture a solution. So when we say that something dissolves, It just means that it goes into an evenly distributed mixture called a solution. Now, like those sugar molecules, oxygen molecules also mix evenly with water molecules to form a solution. In other words, oxygen dissolves in water. But how does the oxygen get there in the first place? Well, there are two main ways that oxygen can enter a body of water, like the ocean or a lake. One way is from the atmosphere, or the air around us. Oxygen makes up about 1/5 of the atmosphere, and these oxygen molecules are constantly bumping into and entering water. Even more oxygen enters water through photosynthesis. You probably know photosynthesis mainly as the way that plants around us feed themselves. But there's actually a whole slew of other photosynthetic living things aside from plants, from tiny green bacteria called cyanobacteria to little phytoplankton with crazy names like dinoflagellate and even crazier bodies. All these creatures can produce food through photosynthesis. But in doing so, they actually also produce oxygen, which then dissolves in the water. What happens to this dissolved oxygen? Remember that like us other living things, the animals, plants, phytoplankton, and bacteria living underwater also take up oxygen to live. Furthermore, oxygen molecules are continually shuttling back and forth between the atmosphere and the water. But note that there's always at least some amount of oxygen that stays dissolved, and this amount depends on water conditions like the saltiness and temperature of water, and how much dissolved oxygen there is compared to the oxygen levels in the air. Now, why does the amount of oxygen already dissolved in the water and the surrounding air matter? Well, it's the same principle as walking into a crowded room from an empty hallway. You don't want to go in, and the people inside are trying to get out. In the same way, if there's more oxygen in the water than in the surrounding air, then the oxygen moves to the roomier atmosphere. Or if there's more oxygen in the air, then the oxygen moves to the water. The principle is the same for the saltiness of water. If you've ever tasted seawater, you know how salty it is, and that's because there's salt dissolved in seawater. Well, those salts are also taking up space in the water. You can imagine that as adding even more people to your crowded room, and the more people there are, the less room there is for you. So the saltier it is, the less space there is for dissolved oxygen. What about temperature? Well, when things heat up, they start moving around. Imagine yourself in a crowded room again. As everyone starts jostling around, it's a lot easier to move out of the room. Again, it's the same for dissolved oxygen. The oxygen molecules warm up and they start vibrating, making it easier for them to escape the water. So let's sum that up. Dissolved oxygen enters water from the atmosphere and from photosynthesis, and then gets taken up by living things or exits into the atmosphere depending on water temperature, saltiness, and the amount of already dissolved oxygen. Now you're probably thinking, who cares what the amount of dissolved oxygen is? Well, remember that living things need oxygen to live, and the more oxygen there is, the more animals can live there. So scientists measure dissolved oxygen to find out how healthy a body of water like a lake or a river is. But animals also adapt to the amount of dissolved oxygen. Fish like salmon need a lot of oxygen so they can only live in colder places, but fish like catfish can live in warmer waters because they need less oxygen. If you've ever wondered why the kinds of fish are different in different places, this is one reason why. Unfortunately, even these fish die off at very low oxygen levels, and humans have been decreasing freshwater oxygen levels in a process called eutrophication. Eutrophication is a big word that just means adding a lot of nutrients good for growing, like nitrogen and phosphorus, to a lake or a river, usually by pollution. That causes a lot of plants and cyanobacteria to bloom. Aren't plants good though? Don't they produce dissolved oxygen? Well, yes, but when they die, they sink to the bottom of the lake and decay there, and this decay uses up all the oxygen in the lake. With no oxygen to breathe, the fish and other animals die and the lake becomes unhealthy. So throw your trash properly and recycle. Together we can preserve our marine and freshwater ecosystems.
Okay, so I really like that video. It's at the end because although recycling is great, it's not necessarily going to help with human-caused increasing the rate of eutrophication. However, the video makes a lot of good points and I want to review them. So the amount of salt and the temperature of the water impact the amount of dissolved oxygen in the water. Oxygen enters the water through the air or by photosynthesis from plants and algae. Too much algae or cyanobacteria leads to low dissolved oxygen levels because when they die, the decomposition takes up oxygen. Nutrients speed up the growth of plants and algae and speed up the eutrophication process. So understanding dissolved oxygen levels in your lake is one more tool to understanding the health of your lake. The DO meter you will receive doesn't look quite like this one, but it will allow you to measure dissolved oxygen and temperature. So we're going to return to that question of why dissolved oxygen and temperature is important. It's important because it helps define where those thermal layers are and helps you classify your lake as warm or cold water lake. And then because of that temperature, it determines what types of fish can live in your lake because DO levels change. And different fish require different levels of dissolved oxygen so that they can live. The dissolved oxygen levels can also indicate if nutrients have the potential to be released by sediment. If you have low DO levels in the bottom of your lake, there's a process by which nutrients are released from the sediment in very low dissolved oxygen conditions. And so that might add additional nutrients to your lake. So this is what the data you will collect will look like. On this side here is the depth in feet. Again, the boat would be up here. So this is the surface water of the lake. On the bottom, we have from 0 to 26 with the dots indicating the amount of dissolved oxygen that was measured at each depth. The triangles indicate temperature measured at each depth. So you'll receive a graphing template like this, or it's also available online. When we look at the dissolved oxygen, we can see that at the surface, it's about 9 milligrams per liter. And it stays around that until about somewhere between 20 and 25 feet deep, the dissolved oxygen level starts to decrease. And then somewhere between 30 and 35, it decreases even further down to 0. So the dissolved oxygen level is very low, starting about 30 feet. With the temperature, we can see that the temperature is about 20 degrees Celsius until we again get to about 20 feet, where it starts to decrease in temperature, and it continues to decrease. So this is our thermocline area right here until it reaches a chilly 8 degrees Celsius on the bottom. So warm water fish need about 4 to 5 milligrams of oxygen, and cold water fish need about 8 milligrams of liter of oxygen. So anything lower than 8 wouldn't be suitable. This area wouldn't be suitable for cold water fish. And then anything less than 4 wouldn't be suitable for any fish. So it can kind of give you an idea of where the fish might be in your water column. So again, just a reminder that that top layer is the epilimnion, that middle layer is the metalimnion where the thermocline is, and that bottom area is the hypolimnion. So this is an example of the individual lake report that you'll receive usually in the spring or winter of following your sampling season. This is for Lakeville Lake in Oakland County, and the blue is the temperature and the orange is the dissolved oxygen, and it looks fairly similar to the results we've been showing you so far, where during the summer, this is August 3rd, the dissolved oxygen levels decrease to 0. There's a pretty strong thermocline. So this is often what we will see in a lake that has the different layers. Here's an interesting, a little bit different of results that some lakes might see. This is Bear Lake in Kalkaska County. It gets to about 60 feet deep, but the oxygen levels stay pretty much the same down to 30 feet. And then there's this weird big bump where the amount of dissolved oxygen really jumps up. And this could be for a couple of reasons. It could be the temperature at that point is also decreasing, so there's more oxygen can be held in the water column. It could also be that at that layer, there might be some algae that really like to be deeper than 30 feet. The light level is just right, and so they're really cranking out some photosynthesis and thus some more oxygen. And because it has a very low nutrient level, that also helps with maintaining a high dissolved oxygen content to the bottom. Okay, so that is the end of the more, I don't know, that part of your brain to learn about the whys and how dissolved oxygen and temperature works. And we're going to move into how our program works here with the CLMP program. So before I move on, is there any questions?
There are no unanswered questions in the chat right now.
Okay. Anybody want to unmute your mic and ask anything? Alexandra? Go ahead.
I have a potentially technical question, and I apologize if I had missed it before. I was curious about if algae and cyanobacteria produce oxygen in the water through photosynthesis, how is it that algal blooms are responsible for creating dead zones?
Good question. And I would say it's that decomposition. So yes, when they're alive, they're creating oxygen. But then, you know, if they're overcrowded and they are fighting for nutrients, and many of them will start dying. And the more you have, the more that will start dying. And so it's that dying that causes those dead zones, because when they die, the dissolved oxygen is getting used up. So even though you have algae there and they're producing oxygen, the rate at which they're dying is larger than the rate at they're making oxygen. Does that help make sense?
Absolutely. Thank you so much.
You're welcome.
I see Jim has his hand up. Jim, do you want to unmute and ask your question?
Yes, thanks. Two things. Part of my lake has a heavily wooded shoreline, and I wonder what the effect of all these leaves falling into the lake are, whether they use up oxygen as they die and dissolve or do whatever leaves do when they fall in the water. And the second thing is we have a weed eradication program. For 6 years, we've been trying to completely eradicate the Eurasian milfoil, and we've been taking a lot of the curly leaf pondweed out of the lake too. So what effect does removing all this biomass from the lake have? Thank you.
Okay, so your first question about the leaves. So first of all, the trees on the riparian area help shade the lake, which is good because it helps it keep it colder, which fish appreciate. You're right, when the leaves fall into the lake, they are going to decompose, and that natural decomposition does use up some oxygen. That's just part of the cycle. But I would say the benefits of having riparian trees to prevent erosion and such is much greater than the impact they're having on the leaves decomposing in the lake. For your second question, one issue with applying herbicides to lakes is that all of that plant matter stays in the lake. And so it is going to decompose, and that decomposition is going to use up oxygen. Does that answer your question?
Yes, what about mechanical harvesting?
Well, mechanical harvesting, you're taking the plants out of the water. So as long as you're disposing of them, not right on the riparian area where all the nutrients are just going to wash back into the lake, that is taking biomass out, and that's not going to decompose in the lake. Okay, so we will move on to the MiCorps program overview. So you have the option of borrowing a meter from us. And what that means is we get that meter to you at the beginning of the season. So some of you picked up meters last week at the in-person CLMP training. Anyone who is borrowing a meter and didn't come last week, you'll be getting an email with instructions of who to contact near you to get that meter. Or there are a few instances where there's nobody near you and you're way far away where I'll be shipping you your meters. Some lakes decide to purchase their own meter. We require a particular kind of meter. So if you are thinking about doing this, please contact us before you do. They run about $1,200 or so. The downside of purchasing your own meter is that you are responsible for the maintenance of the dissolved oxygen meter. So every year when the meters are returned to me, there's some maintenance that we do to make sure the meter's in good shape for the next year. If they're broken, we might send them to get fixed at one of our at the distributors. And that would be on your own, but you'll have access to your meter and you don't have to borrow one from us every year. You're going to measure dissolved oxygen and temperature twice a month, starting this month, May through September. So we have two types of monitors that you might get. We have a few of the older model, which is the YSI 550A, which is here on the left. They still work great, and we want more people to be able to participate. So we do have some of these meters still available, but the majority of our meters are this other model called the Pro 20. This is just some real world examples of each of the meters. You can see the meter. There's the long cable that will go into the water column. There's on this particular one might be a deep lake. There's a weight to help keep it steady and straight. And this is the actual probe. With the other one, again, here's the cord. Here's the probe right here. So this is a close-up of a probe. There's a plastic guard on the probe, which I'll talk about in just a second. And then there's a membrane cap that actually is what measures the dissolved oxygen as the water passes through. And then there's a little tiny silver nub that is called the thermistor, and that is where the temperature is measured. So the probes look a little different for each of the meters. For the 550As, the membrane itself looks the same for each type of meter. But for the 550As, the cage that protects that very sensitive membrane is a black plastic piece. And in the Pro 20s, it's a heavier metal piece. And then to go over the membrane to keep it moist, we have these covers. This one's clear, this one's not. Both of them have little pieces of sponge in the bottom of them that are to be kept moist, but we don't want this cap area to be full of water. This is an example of the kit you will get if you're borrowing one of our meters. It includes some type of storage box. Some of them look like this. These are our new ones that we just ordered this last year. It's a heavy duty box so that you can protect that very expensive piece of equipment. Some of them are more like Rubbermaid types, and we just ask you to be careful not to drop those boxes. You'll get the probe and cable along with the meter. Most of them are 100 feet long. We have a few lakes that are more than 100 feet, and we have some spools that are 200 feet in length. You also will get some spare batteries for the meter. Very rarely have I had to change batteries. They just don't take up that much energy. You'll also get a quick start calibration card, which we'll go over calibration here in a minute. And you'll get a reminder that this is the property of MiCorps and EGLE, and that we need you to return it at the end of the year. You'll also receive some extra dissolved oxygen membrane and electrolyte solution. And then you'll also receive an agreement letter that you will take care of the meter and that you'll return it at the end of the year. So to prepare for sampling, you want to make sure you have calm and dry weather, just like Eric mentioned for phosphorus and Secchi. You want to pack up your equipment, including your safety equipment, and bring a friend to help with data recording. Check your quick reference procedure checklist. Make sure you have all your data forms. Connect your cable to your meter. If you have a Pro 20, those cables can detach. They do not detach for the 550As. And then most importantly, in addition to all the other important things, turn on your meter for 15 minutes before you start calibrating. They need to warm up and to stabilize before you start calibrating. So here is a video that's available online of how to calibrate a Pro 20. This video is to show you how to calibrate your YSI Pro 20 dissolved oxygen and temperature meter. It's really pretty simple. First, you want to check inside the cap that covers your probe and make sure that the sponge that's inside is wet. Always want to keep the environment for the DO probe moist, but it shouldn't be standing water. You just put that back on and then turn the unit on. You'll see at the top here, there's two measurements. One says milligrams per liter, mg/L. One says percent. The percent is the percent saturation, how much oxygen is in this container right now. And then the milligram per liter is what you'll be measuring when you put the probe into your lake. The temperature units are in degrees Celsius, which is what you want. And to calibrate, really all we have to do is hit this button here, press it, and then hold for about 3 seconds. And then it will say calibrating percent dissolved oxygen until it says calibration successful. And then you're all set. Notice it did go away, the calibration successful. So if it's taking some time and you look away, you might miss that. But if you wanted to double check, all you do is hit calibration again, hold it, calibration successful, and you're ready to go. Have fun. So that's how you calibrate a Pro 20, and there's about a half dozen of you that have the 550As, and this is how you calibrate the 550As. Hello, this video is to show you how to calibrate your YSI 550A dissolved oxygen and temperature meter. If you have a YSI Pro 20, there's a separate video for that. The first thing to do is to turn on your unit and then let it set for about 15 minutes. I've already done that. The next thing you want to do is make sure that the sponge inside your probe cap is moist. There shouldn't be standing water, it just needs to be slightly wet. If it's dry, make sure you put a couple of drops of water in there and then replace the cap. The next step in calibration is to hit both the down arrow and the up arrow at the same time. When you do that, you will get CAL for calibration. You then hit enter. Once you hit enter, a number will come up. This is where you will enter the elevation of where your lake is. In the Lansing area, the elevation is about 900 feet. So there's a 9. However, if I was somewhere else and the elevation was, let's say, 700, I would put the down arrow twice to get to 7. If it was 1,000, I would move it up a couple to get it to 10, and so on. Once you have the correct elevation, hit enter again, and your unit will begin calibrating. It'll jump around quite a bit. Mine is not, because I have had this meter on for a while and it's had time to stabilize already. So you can see it's just jumping between 0.2, 0.1 to 0.2, 94.1 to 0.2. Your percent saturation should be between 93 and 98%. If it's not, keep waiting, and if it still does not, then you'll need to turn it off and try again. But since we seem to have a stable number, and it is between those numbers, I press enter. The next thing that comes up is the percent salinity. We have fresh water in Michigan, so we don't need to worry about this. So just make sure it says 0 and hit enter again. And there you go. You're all set. Before you go out and measure dissolved oxygen in your lake, however, you need to change the units from percent saturation to milligrams per liter. And you do that just by hitting the mode button once, and it changed to milligrams per liter. I see it's in degrees Celsius, which is what you want for the data sheets that we give you. If it says degrees Fahrenheit, all you need to do is hit the mode button and the down button at the same time, and it will change to Fahrenheit. Hit it again, both the arrow, down arrow and mode, and it will change back to degrees Celsius. Now you're all set. Have fun measuring your dissolved oxygen in your lake. Okay, so those videos are available online, so refer back to them as many times as you need to throughout the monitoring season. So there might be cases, and this happens very infrequently, but there may be cases where you cannot get your meter to calibrate. And in that case, you might have to replace that dissolved oxygen membrane. In this picture, it's gray, but for all of you, it will be yellow. So as I mentioned earlier, you will have either some kind of container that will have some tissue paper in it, or in this case, it was some cardboard to hold it in place, and you will have an extra membrane. You'll also receive some of this solution that needs to be put into the membrane in order to make the membrane be able to conduct electricity with the meter. You will only need to do this if the monitor will not calibrate. You can throw out the old membrane if you do replace it. And if you have questions, just go ahead and call me right away, and I will get back to you as soon as I can to help you out. There is also a how to replace your membrane video online, but for the sake of time, I don't think I included it here unless it surprises me in a minute. But there is a video online on how to change your membrane. So to get your sample, you're going to proceed to your sampling location. You're going to anchor just upwind of that deep basin again and drift back over that deepest spot as you do for all the other parameters. You're going to check the depth to make sure you're at the right spot. You can do that with a depth finder or a weighted line, or use your Secchi disc meter, the rope that you have to lower it down. You'll have to turn on your meter again. They do turn off after a certain period of time. Once you have, as long as you have calibrated it, you don't need to calibrate it again. As long as it's been done the same day, like right before you went out. You'll take your cap off, but leave that guard on it to protect it. And then you'll begin taking measurements at 1 feet deep for the first measurement. This is what your data sheet looks like. It has the standard information that you need to put at the top, where the lake is, what your field ID number is, who's doing the sampling, and the date and time. The weather conditions, and the sampling station depth, just to make sure you're at that deepest point of your lake. Circle what type of meter you have, and your meter ID, which is a number on the outside of your meter box, and it's probably also written on the meter. For the 550A, we need to make sure that you did all of the calibrations. So we want to make sure that you check the percent air saturation, which I noticed on the video it said between 93 and 98, but it's actually between 93 and 103. And then if you can't get this value, you need to troubleshoot and figure out if you need to replace the membrane. And then the lake elevation value. If you have a 550A, I just earlier this morning sent you a list of the elevations for your lakes. You're going to draw a quick diagram of your lake and where the deepest point is. And then this is just how you take measurements. So as I noted before, you're going to start at 1 foot deep, and you're just going to be moving the probe kind of like bobbing up and down motion. And that's just to make sure the water is flowing over that probe. And as I mentioned, the DO reading will probably drift a little bit. And once it slows down enough to, maybe for this example, it's 5.6 to 5.72 to 5.5 to 5.8 to 5.7. Just try to take what it seems to be landing on the most. And then you'll go to the next depth on your data sheet and continue to do so. But you want to make sure that your last measurement is about 2 to 3 feet above the sediment, just to make sure you protect that probe and don't get it into the mud or into the sand. If that does happen, like, you'll know, because when you bring it back up, it'll probably have some debris or mud on it. Just swish it off in the water to clean it off, and then you can put it back down. Just make sure you make it more shallow to get that last measurement. So this is what your data sheet will look like, where you're going to enter all of your information. Note the temperature is in degrees Celsius, and the dissolved oxygen is in milligrams per liter, not percent saturation. So, you're going to be measuring at 5-foot intervals, except for there is area where you're measuring every 2.5 feet, and that's an attempt to get at that thermocline. If you have a really shallow lake, you can measure more often between this 1 to 15 range. Just make sure you note that on your data sheet in some fashion, maybe put them over here. And I believe when you enter data, you can enter any depth and the measurements that you took at that depth. So again, this is what your data will look like after you graph it. And if you need help graphing it, if you can't figure out how to do it, just contact somebody from the MiCorps team. And then just like all the other data sheets, it has some information to remind you to submit your data online and to do so by October 31st. So these are some end of the year, don't forget for one thing, you need to return the meter, and that needs to be done with the last water chemistry sample drop off. For those in the UP who want to measure their dissolved oxygen further into September and your drop-off dates are late August, just please contact me so that we can make arrangements on how we'll get that dissolved oxygen meter to me. If you do forget, or please again call me or email me so that we can make arrangements. But if at all possible, please drop them off with your last water chemistry sample, your summer phosphorus sample. Remember that the sponge inside the container should be damp, not pooled up. They'll come damp. I've just checked them all this last week to make sure everything is ready to go, your membrane's on, and that your meters calibrate. And then just a reminder that videos are available on our website. And with that, I'll take some questions and just reiterate that if you have any questions regarding your dissolved oxygen meters, to contact me at any time.
Thanks, Tamara. We did have a few questions come in the chat, so we'll start with the first one. Do you wet the sponge with deionized or distilled water?
No, nope, you don't. It can be the lake water, it can be tap water. I have access to deionized water, and that's not what we use for just keeping it wet.
All right, great. Next question. Do you have enough meters for all those who need them?
We do this year. So the last couple of years, I think, the last 3 years, like I've been able to come up with just enough. So we have this year 41 meters available. But my understanding is that in other years where we have too many people that would like to do it, that more people that want to do it than we do meters, we will people will share meters. So if there's a nearby lake, people will share meters between the two of them. And even this year we have one lake where the lakes are super close together. The lake associations are very well associated with each other, so they only have 1 meter between the two of them.
Perfect. Thank you. Next question. How do I do depths when the deepest part of my lake is only 9 feet deep?
That is a good question. And that's I hope maybe I answered that after you asked that. So on that data sheet, I would just do right at the surface, not even a foot deep, and then go a foot and maybe a foot and a half. You could go every half foot or at least every foot to get different measurements. And as I said, that's not on that data sheet, but you can just make a note, just make, your own columns. And then when you enter that data online, there's the opportunity to put more measurements than is on the data sheet.
Thanks, Tamara. The next question is from Doug. How would I know when I'm just a couple feet above the lake bed?
Well, that's why it's important to make sure when you get to the deepest part of your lake, that you either have a depth finder that tells you the depth, or that you use your Secchi disc with the meter tape. That was the word I was trying to think of earlier, the meter tape, or the, isn't feet, the tape that goes down so that you can measure that depth. So if you measure it and you measure 25 feet, then you know that around 22 feet or 21 feet, you need to start being really careful because you know you're getting close to the bottom.
Great advice, Tamara. All right, Mike, I see you have your hand raised. Would you like to unmute yourself and ask your question?
Okay, am I unmuted here? I think I am.
Yes, we can hear you.
All right. We enrolled our lake, Big Shag Lake, in Marquette for the first time last year. We did the Secchi disc, and this year we're going to do the Secchi and the phosphorus testing. My question, since we just enrolled it for the first time last year, we saw the report that you guys generate for the lake. And my question is about how like dissolved oxygen and other tests in this report get integrated with perhaps other agencies in Michigan, like the DNR. And the reason why I ask this is that, obviously with all we did was the Secchi disc last year, so there isn't a lot of information in the report, but there is some older Secchi transparency reports from decades ago, that are in that report. And I know a couple of years ago, we got a presentation from the DNR on the survey on our lake, and it had in that presentation the dissolved oxygen results for summer and winter and fall and over a couple of 3 years. This is John Bauman that did the presentation for us from the DNR and the UP. So my question is, do you integrate, so like next year, we're going to figure out what tests we want to do next year, whether it's dissolved oxygen or chlorophyll or whatever. So my question is, whether you work with the DNR or you can incorporate the DNR dissolved oxygen results into this report, which, we'd like to see.
Well, I can answer part of that question as far as our data and how it's used. Our data is used by the DNR. They do look at our data. They use our online database, and they do use that dissolved oxygen temperature data. And in fact, they encourage, they want us to have as many meters and people doing this as possible because they do use that data. The DNR data does not go into our database because they might do things a little differently, not take it at the same depths, things like that. And they're not the MiCorps program. So that information doesn't go into our database.
Okay.
Did you want to add anything, Joe?
I think that's totally accurate. Yeah, our reports will just have the MiCorps data, but our team works really closely with folks at DNR to make sure they're aware of the data, and John Bauman knows about this program, so they have the access to all of this data as well.
John was very excited that Big Shag had volunteers on it because he wants more data, and just like we've been saying, there's not a ton of us. And so having folks like all of you, and then Mike, he was excited that there was more data being collected. So he does look at it.
Okay, great. Thank you.
All right. As far as questions in the chat, I just wanted to note to folks that Marie had asked for recommendations on depth finders. We through MiCorps don't have any particular equipment that we recommend, but if anybody has a depth finder they like using, you can of course put that recommendation in the chat. This is a great opportunity for sharing information among our volunteers.